Good morning, students. DNA supercoiling. What is DNA supercoiling? Supercoils are twisted three-dimensional structures and which are more favorable energetically for transcription as well as for translation. So this is the advanced stage of DNA molecules within the nucleus, that is supercoiling. So we are going to discuss supercoiling of DNA molecules. As you know, DNA is a double-stranded linear structures, but this is not the relaxed state of this DNA molecules. It is sometimes found in the cells in the form of a circle, to which we call the supercoiling. This topological state of supercoiling is the next highest order level of DNA organization after the linear relaxed state. In order for a DNA molecule to be supercoiled, it must be in a closed loop or in a circular form, and their ends should be fixed, they should be attached with each other. Closure is achieved most often by joining the ends of each DNA strand from a circle. That is the circular DNA, that is the DNA molecules are being attached with these two strands if they are coming. So they should be attached with each other in the form like this. That is the circular DNA. This is our not only the way to close a loop in eukaryotic chromosomes. There are some other methods also. Loops are formed by holding the ends on a protein scaffold pattern or conformation of DNA molecules, as I have discussed in earlier lecture, that there are the various patterns of the DNA conformation. So this is occurs with the help of this scaffold pattern of conformation of DNA molecules, that is supercoiling. Now the question arises, what is the need or why these DNA molecules are being supercoiled? So many naturally occurring DNA molecules are circular, found in nucleus. Without, there is no free end of 5 and 3 ends, and these two ends they attached with each other. Here should be noted that always the 5 end of one strand can only join with their own 3 end strand. The DNA molecules, as you know, that it is a double stranded. Suppose this is double stranded, this one is double stranded. If it becomes circular, then this 5 end, then this 5 end of one strand will join only the 3 ends of the same strand, not with this one or with this one. It becomes circular. It is compulsory. And another importance of the supercoiling is that DNA molecules, as it is a very long and nucleus is very small, so it resides inside the nucleus in the compact form. And another thing that it stores the potential energy before the replication and transcription it requires. And if we remove the supercoiling by protein binding, it puts the molecules under stress. And this stress only can be relieved by separating the strands. Here you just see the example. Suppose you are having the two strings with each other and you are just coiled them with each other and this. So just to relieve them from the stress, what is the process? We have to unbind it. But when we will unbind it, it will put more stress at the lower circles. So stress the two strands are having with each other and only can be relieved by separating these two strands or unwinding these strings. So different genes have different optimal levels of supercoiling. The coiling may be the tight or it may be the loose. So it all being affected by the gene expression. And the gene regulation play an important role. Usually one negative supercoils with the 20 positive supercoilings. In the next slide, we'll discuss what is negative supercoiling and positive supercoiling. In this slide, you can understand that the DNA molecules, the two strands, if they become circular, that is covalently closed circular template, that is the supercoiling of the DNA or the circular DNA. When such type of DNA will replicate and translate, then what will happen? Just to relieve the stress, here you can see the unwinding of these molecules occurs. And as soon as the unwinding occurs from one end, it will put more stress on another ends. Here the supercoiling or they are becoming more tightly twisted. That is overwound regions, because from here we are relieving the stress. Is it clear? So this is the pattern of that how the DNA molecules are being supercoiled and they relieve their stress. This is the supercoiling stage. It was the supercoiling stage of the DNA molecules, and this structure is energetically favored over one containing the unbound loop. Why? You just note down in the earlier picture you have noted down that the supercoiled forms produced from unwinding the DNA is right-handed. This unwind product, if it is right-handed superhelix, is called negatively supercoiled. It is called negative if it is right-handed. It's right-handed, that is there are the two hands. The supercoiling suppose occur in this way, like you can understand like this here. This one. This is the supercoiling. This it may be the another, and another thing may be by this pattern. Is it clear to you? One is right-handed and another is left-handed. Right-handed, this one is right-handed supercoiling. So it is known as negatively supercoiled. And if it is left-handed, this one is left-handed, then it is positively supercoiled DNA molecules. Is it clear to you? Negative and positive. However, mostly the DNA molecules in organisms are found to be negatively supercoiled, that is right-handed. Maximum amount of the DNA molecules is supercoiled negatively. So is it clear to you? Supercoiling of DNA negative and positive strain. Again, with the help of this slide, you can understand the positive supercoiling and the negative supercoiling. If the turns of the DNA helical structures occurs in a right-handed way, then the supercoiling is negative. Here in this you can see. This is the turn at right hand. This is negative supercoiling and positive supercoiling of DNA is clear to you. That if the turn occurs at right hand, then it will be negative, and if the turns occur at left hand, then it will be the positive supercoiling of DNA molecules. As you know that B conformation of DNA molecules have 420 base pairs, that is adenine, thymine, guanine and cytosine, and these 420 base pairs they make a turn after the 10 base pairs, that is 10.5 approximately. So if it occurs, then how many turns will be there? There will be 42 turns if it occurs after 10 base pairs. If we join the ends of this helix, then this circular DNA will is said to be relaxed state. Clear? Suppose now we unwind this double helical circular DNA and we unwind the 6 turns, then how many turns will remain? That 42 minus 6 will be 36 turns. And this conformation of DNA will be bDNA. It can adopt a structure with 36 tons of DNA molecules, and again it will be a superhelical structure. The unwinding is taken up by allowing the previous unwound region to adopt the B conformation of DNA. There will be no change in the length of DNA molecules. However, the linking number will be changed. Now the linking number of the DNA will be, except 42, it will be 36 and the tone will be changed. And we mathematically represent all these linking and twisting and worth number of DNA molecule with the help of equations we will discuss further. All this function occurs with the help of enzymes. And these enzymes were first of all discovered by Gellert and Wang in 1970. These enzymes are called top isomerases. That is the topology of the DNA, and it occurs with the help of enzymes to which we call top isomerases. And these top isomerases, first of all, uh, coined by Gilbert Wong in early 1970s. And these are the enzymes which regulate the supercoiling of DNA molecules within the nucleus. They are called isomers because they are similar isomerized. They are the isomers of each other. Hence, we call them top isomers. There are two types of top isomers in DNA topology: top isomers first and top isomers second. The top isomers first, it relieves the supercoiling. What does it do? It relieves the supercoiling of DNA molecules. And by releasing the supercoiling or unwinding the DNA helix, the DNA molecules is being relaxed. And these enzymes, they do without the input of energy, which were top isomers 1. They act by cutting one strand of the supercoiled DNA helix and rotate one strand about the other and then release the strands. Suppose this is the supercoiling of the DNA, circular state, it is like this. So when unwinds occurs, it will cut from here, these two joints, and it will be released. It will become like this, right? It will become like this, supercoiling of the DNA. And this is the all this function done by the top isomers first, and it needs no energy. It relieves or it uh, cut the two supercoiled DNA molecules and it relieves the strands stress. This reaction proceeds in a full mode, and however, the another top isomers, it which cleave the DNA molecules from a covalent bond, it occurs usually at the point of tyrosine bond, where the tyrosine is being attached with the help of phosphodiester bond, and it releases the linking energy. Clear? Now the top isomer second, it function to put supercoils into the DNA. It's just opposite to the top isomer first. First it cuts the DNA molecules and relieves the DNA molecules stress. Whereas the top isomer second, its function to put the supercoils into the DNA. The one more enzyme, that is enzyme gyrase, it is required for DNA to replicate, and that is the type second top isomerase. The gyrase enzyme is required for the replication of DNA molecules, and this is occurs in presence of the top isomer enzyme second. The introduction of supercoils into the DNA requires the hydrolysis of ATP molecules. It requires energy. Top 2 isomerase, it requires energy because now the replication and translation starts. Top 2 enzymes function by making the double strand breaks down into the DNA molecules, and it rotates one strand to the another strands and again rejoining them. Hence, it requires energy into the form of ATP molecules. Since supercoiling is an active process, it requires energy, and this indicates the supercoiled DNA is a high energy form of DNA molecules. That's why I've said that this is the favorable energetically process, that is supercoiling of DNA molecules. Now, what is the significance of these two top isomerases, isomer first and isomer second? Top isomerases are the targets for a number of important anti-cancer drugs. These top isomerases are used to treat the cancer patient. Top isomerase second targeted the drugs are used in approximately half of the chemotherapeutic treatments. Top isomerase first is the target for a number of promising agents, including compactactin. It is a drug name of the traditional name of the drugs used for the treatment of cancer. Because these top isomers, they target the DNA molecules of the cells which are dividing and dividing so rapidly. What is the role of this top isomers? Because it stops the replication and translation of DNA molecules. So these isomers, they are used for treating the cancer patients. And first of all, this process was discussed by Monori in 1966. Dr. Monori Evol and Mansuk. It was not until 1985 that another group of researchers determined that compactactin is a top isomerase first poison. So that's all about the supercoiling of DNA molecules. And this slide is just showing you the difference between the top isomer first and top isomer seconds. These are the human DNA top isomerases. So the first DNA isomerase, its type is 1B. And DNA cleavage occurs at this is the single strand base pairs. It relaxes both negative and positive supercoiling DNA, the role of this first top isomers. And its function is replication, transcription, and recombination of DNA molecules. This SSB is single strand base pair. It cleaved, it breaks down the single strand break pair. Now the third type of the DNA isomerase is the third A, third B, second A, and second B. And this type is also first A, and it also breaks down the single strand of base pair, and it relaxes only negative coiling of DNA molecules. Negative, that is right-handed. Recombination and function is recombination, transcription, and ribosomal RNA. Now the third B, it is also a single strand base pair, and it leaves only negative strand, negative supercoiling of the DNA, and its function is recombination. Second A, it attacks on both the strands of DNA molecules, that is double stranded base pairs, and it relaxes both negative as well as positive supercoiling of DNA, and its function is condensation and replication inside the nucleus. The second one, second A, it also attacks on the double strand base pairs of the DNA, and it also relieves or relaxes both negative and positive supercoiling of DNA. However, its function is not so clearly defined in the. Now, how is the linking number in twist? And we have to just calculate. The linking number is being denoted as LK of the DNA molecules. The linking number is equal to twisting and volts of the DNA molecules, where TW is being denoted as a twist, is a measure of the helical winding of the DNA strand around each other, and width is a measure of the coiling of the axis of the double helix structures. Suppose the linking number is 42, and how many terms it will accompany? That is twist, the linking number will equal to twist, that is the 42 divided by 10. So it will be 420 divided by 10. So it will be the linking number of the DNA molecules. So we denote this linking number of the DNA with this equations, that is LK is equal to twist and fourth number of DNA